Welcome everyone. I'm Michelle Lafoy, and Melissa Petrich is also here. If you have any questions, feel free to put them in the chat, and she can interrupt me and ask questions as we go along. Okay, so today we're going to go over our protocols that are in the revised SoundToxins manual that was released in 2023. If you don't have a copy of this, hopefully you'll be able to get one at our in-person meeting in 2 weeks from now on the 18th and the 19th. It's also available online. And today we're going to cover field sample collection, which includes collecting environmental data and how to collect the whole water and the net tow samples. And then sample processing back at the lab, including live microscope identification, archiving samples, and how to send alerts, as well as data entry and how to do that. So for field sample collection for our program, these sites are near shore sites, either from a dock or in some cases from a boat, where a few of our sites are located close to shore, but folks take boats to get them. Our sampling is weekly from March to October, and then from November to February, sampling is every other week. Some sites do year-round sampling, and that's okay as well. And sometimes if there are active blooms happening because the timing of blooms has changed from what we historically knew to be more frequent in the winter time than they used to be, we might ask specific sites to sample a little bit longer, more frequently in the winter as well. And then most importantly, only when conditions are safe for sampling. Today it's quite snowy and icy in a lot of our sampling locations. So on a day like today, you might want to exercise a lot of caution. Make sure you know what the environmental conditions are before you go out, and make sure you're only sampling when it's safe to do so. And as I mentioned, what you collect when you go to your field site includes environmental observations, one whole water sample, and one vertical net tow water sample. So when you go to the field, you should bring with you a bucket, sometimes useful to put all your other equipment into that bucket to carry it. A plankton net, we use 20 micron mesh size plankton nets. Refractometer, this is to measure salinity. Some folks will leave this item back at the lab, and that's okay because the salinity won't change much between your sampling location and the lab, but they are hardy enough to be brought out into the field if you would like. And the thermometer to measure air and water temperature, a 2-liter plastic bottle to collect your whole water sample, a glass jar that's used to collect your net tow sample, your data sheet, and something to write with. So the environmental observations we collect in the field on our data sheets include the time, the date, and the sample location, especially if you sample multiple locations, make sure you write that down. For air temperature, we'll hold our thermometer in the air before it gets wet for 1 minute and record the temperature in degrees Celsius. For wind, in our database, we use a drop-down menu to select between the options of none, light, moderate, or strong. Some folks might have weather stations near them that they might consult, and they'll have wind speed there, but you can just translate that into one of these categories for your data entry. And for weather conditions, we're mostly looking for basically how much cloud cover there is. So whether it's partly cloudy, fully sunny, fully cloudy, or overcast. And again, these are categorical on our database. And then for the tide, we are also looking at just whether it's high, low, incoming, or outgoing. And so you can check a local tide chart to see a location close to your sampling site for that, and then record that as well. So our whole water surface is a surface sample. So we're just capturing the surface layer of the water. The way we do this is by plunging the bucket through the surface of the water to fill it. You don't have to go super deep, just as long as you're grabbing water from the surface enough to fill the bucket. pull your bucket up carefully, and then to measure the water temperature, place the thermometer in the bucket for 1 minute and then record the temperature in degrees Celsius. For salinity, again, you could do this either at the lab or in the field, but if you're doing it in the field, what you would want to do is take a drop of water and put it on the viewing chamber of your refractometer, and then place the little flapping part down to close the lid. And then look through the eyepiece like towards a bright area, but not directly at the sun, so that you can look through and see the value. And what it'll look like is there's a scale inside your refractometer, and the half of it will be blue and half of it will be white. And where that line reaches, that's the number you want to record. And then from that bucket of water, we want to fill our 2-liter plastic bottle of water to bring the whole water sample back to the lab. And then to clean up, you simply want to rinse the bucket, the thermometer, and the refractometer with fresh water. You don't want to leave salt on any of those items or salt water. And then you can let them just air dry. For the vertical net tow, the way our nets are we have our 20 micron mesh, as I mentioned, that has a cod end at the bottom. And then these are usually tied to a rope or a string. Sometimes our ropes, we recommend like marking on them like each meter of rope from the surface or from the top of your net. So that way we can get an idea of how much, how deep and how much water you're filtering. So the way we will do this is to lower the net slowly but consistently through the water. Let it drop basically with the weight of the cod end. Once it's filled up with water, it'll sink to the bottom. And then you want to stop when the cod end reaches near the bottom. So we want to get as much of the vertical depth of water as possible, but we also don't want our net to get caught on rocks or sand or anything that might be at the bottom of the water, because they're very delicate. So we don't want it to like drop all the way to the bottom, but when the cod end reaches the bottom, that's when you want to stop. At this time, you would then start pulling the net back up, and we say at a steady pace of about 1 meter per second, just consistently pulling the net through the water as vertically as possible. And then what you want to notice is when you stopped, where the rope is hitting the surface of the water. And then you want to count how many meters below that water point you have collected water. So having markers on your rope can help. And also some folks will, like when you're pulling it up, if your rope is dry to begin with, you can like feel when it starts getting wet, depending on how close you are to the water. Some folks are right on a floating dock, so you're really close and you can tell where the top of the water is, but some folks are off kind of like at a raised dock, and so that makes it just like a little bit more challenging, so just pay attention to that.
And then hey, Michelle, we had a question come in about that 1 meter a second seems kind of fast, and they usually bring it up at the same rate that it drops. What are your thoughts on that?
Good question. So this whole process, we will end up, the reason we write down the depth of the rope and to estimate how much water we're sampling is to do what we call a semi-quantitative calculation. So for just our target species, and we'll talk about this later, we'll do cell counts to estimate how many cells are in the water. It's definitely a semi-quantitative calculation because there's a lot of variables that are not accounted for. So if it's a particularly, the waters are rough or there's a current or something, if you're pulling your net through the water, it doesn't matter what pace you're pulling it, because a lot more water could be going through the top of the net than if the water's perfectly calm and still. So this is one of those areas where we are kind of trying to estimate, like figure out the best way to get some information out of our sampling methods, even though we know that they're not perfect. So I think 1 meter per second is what has been the protocol for that's been written down. And I also think that that's a little bit hard to estimate. Let me think. So I usually, so when I do it, I don't know if I'm actually getting a meter per second. I don't know if I've actually measured it myself. But I think what's important is that you pull it up kind of at like a steady rate, but not too fast, but also not too slow, because or else you're just letting like a lot more water like kind of go in and out. So this might be a good question that I will consult with our some scientists, our scientific committee that we're forming to give feedback on, and we might adjust this. If you, I don't know who asked that question, so I'm not looking at the chat right now, but if you've been doing it, I think what's most important is that you're consistent within your site. So if you're the person doing it and you've been doing it the same way, there's no need to change. And if you have a larger group that all sample at the same site, kind of try to align with your fellow samplers to do it in a similar way, because again, it's not perfectly quantitative, but what we're looking for is kind of like relative consistency. So if you sample 1 week and then you sample the next week doing it the same way, and you see like a rise in cells, we won't know exactly how much more cells there are, but we can say, oh, it seems like there are more at this site, and that's something we should pay attention to. And if you include in your environmental conditions, like which way the tide's going, and in the comments, if there's like a really strong current or some other variable that seems wildly different from week to week, then when you send us an email saying, hey, we saw a lot more, let's say, for example, Alexandra met our site this week than last week, and it seems like a really big jump, we can also say, oh, okay, well, there's a really big jump, but also your current was really strong, so you might have been pulling in more, but we still are going to pay attention to that, but at least we have that additional information. So this is why the environmental conditions are also helpful to record. And of course, you guys all know your sites better than me because you're there every single week. And so when you see something that's different or amiss, and if you communicate that to us, that's really helpful as well. So if you say like, okay, I saw a huge amount of Alexandrian, but I also know that like our sampling was slightly different this week than last week, it can help give us more information on like how concerned to be. But generally, if you're sending an alert, we're always going to be like paying attention to that. So that's more of like if you have a drop in cells, but you know that the sampling was different, we might say, okay, let's not take that at face value. Let's keep looking at that site. Does that help?
Great. Yeah, this was a question from Paulette. So I think that answers it. But also maybe we can talk about it more at the in-person training and see, if we want to be everyone doing 1 meter per second if we can, or if we want to consistently go about what we've been doing at our sites.
Sounds good. And yeah.
And they have another question, and also Paulette, you can feel free to speak up, but they're wondering about if you need to jiggle the net as it comes out of the water to dislodge plankton that are stuck to the side of the net. That's what they do. So
yes, great question. That segues really well into what I was about to say right after this. But just to the other thing I was going to say was thank you for calling out these like kind of small, they're not small questions, but like specific questions, because we are refining our protocols year to year and working on improving them. So we definitely want to make sure that things are like consistent and like scientifically accurate moving forward. So we are like re-looking at these protocols, even though they've been around for a long time and have served the program well. We're always looking for ways to improve them. So thanks for calling out those questions. And yes, so once, as you are pulling your net out of the water and you get to the point where you start to see the net, you want to look at it and assess. So the nets are generally, they're white when they are brand new, but of course over time they might get slightly colored, even if you clean them really well and take good care of them, but they're still like light colored. And what you want to look at is the color of the water against the net as you're pulling it up, to assess whether you're going to repeat this process or if one pulls enough. So if you're, if the conditions are that the water is full of phytoplankton and stuff, as you pull it up, the water will look colored against the net, and you might know like, okay, that's enough. I'm going to have plenty of phytoplankton to look at here. But if the water still looks very clear and the net looks very clean, then it might be that you don't have a lot of abundance in your water at all. And in order to make sure that we're like kind of catching everything and seeing what's there and being able to assess, you might want to repeat the pull up to 3 times after the first tow. And when you're pulling up the net, if you are noticing that it's really gunky and there's a lot of stuff stuck to the edge, you can, swirl the net a little bit in the water. In theory, once when you're pulling it up out of the water, and if you can kind of like wiggle your rope a little bit, it might help take some of the plankton off the side of your net and swish it into the cod end, which is where we want it. And I think this is one of those places where in an ideal world, we would use like filtered seawater and like rinse down the side of the net to make sure that we're capturing everything, especially the things that are more prone to sticking. But again, so the protocols were made to be like kind of quick, rapid, reproducible, and easily spread to a large network. Not everyone has access to like filtered seawater that they keep in like a sterile jar to rinse down the net. So we do this as well as possible by just swirling the net in the water and getting the water down, getting the phytoplankton down the edges. So then we're hoping to get a good amount of phytoplankton in your cod end by the end, so that when you look at it under the microscope, you're seeing a good representation of what's in the water. That's the goal. So if you do end up pulling the net through the water more times, then just record that, because you're going to multiply the depth by the number of times to get the total distance towed. From there, another question.
We have another question. Quick question that came in from Doug, wondering when you say like a big jump, so like one sampling to the next sampling, what type of like order of magnitude are we talking about? Or is it like doubling? Like, I'm guessing we'll cover this a little bit more, but what would look concerning?
We are going to cover this a bit more, and partially this depends on the species. So we'll talk a little bit more about this in this session, and then we'll talk about it a lot at the in-person training on the 18th, which will also be virtual for anyone who can't make it in person. And so it depends on the species, what like counts as what's concerning, and what counts as a big jump. So for Alexandrium, for example, we know that even low cell counts can have a lot of toxicity. So even if you have like really low cell counts, we would still like want to know about it and might pay attention to it. But for other species, for example, Pseudonychia, if you have that in your water in a large abundance, it could be a concern. But for most of the sites in the SoundToxins network at the moment, the Pseudonychia doesn't produce domoic acid as much in our waters. And so if you just see a couple of cells, it's unlikely that they're producing enough toxin to be a concern. But if you're seeing kind of like a large amount of them, then it would be more of a concern. And anyway, so we have an alert chart that we'll share with you all that tells you kind of like guidelines on when to send alerts in. And I think like a big jump from anytime you're seeing like order of magnitude bigger or tenfold increase, like that's a big jump. Like you see one cell 1 week and then you see 10 the next, like 10 still might seem low, but like that's a bigger increase. And if you see 10 1 week and then you see 100 the next week, like that's pretty significant. But we'll give some more guidance on that later in the talk. Okay, I think we're at the stage of filling. Wait, is there any other questions? I think we're at the stage of filling our glass jar. And so here's another instance of where what you want to do is unhook your net from your cod end and swirl again, because things can stick to the edges of the cod end gently, because you don't want to spill too much of this liquid at this point. So you swirl it gently and then transfer it into the jar. And be careful to not spill as much as possible, because again, we try to measure this volume for our calculations later. So just note that at this time. And then that's what you bring back to the lab. And then for the cleanup for the field portion here is that you want to rinse the net again in fresh water really well, especially because it's a very fine mesh. And if little tiny things get caught in it over time, that can like affect our sampling. So you want to rinse it with fresh water and then hang the net out to dry out of direct sunlight, because again, it's such a fine mesh that the UV rays can damage it and cause bigger holes than we want. Okay. So next we'll talk about the processing back at the lab. So the supplies that you need for this include, realize that a microscope's not on there, but that's the most important thing. But other things that you'll need are a Palmer-Maloney slide, a graduated cylinder to measure your volume, a 50 milliliter glass tube that comes with a screw top, pipettes, your net toe sample, your whole water sample as well, scintillation vials for storing our archive samples, formalin for preserving our samples, and still your data sheet and your pencil to continue recording what we find. So we have two processes for the net toe sample and the whole water sample that are similar but slightly different. So for the net toe sample, the steps that we're going to follow are to look at the sample live under the microscope, and we're looking for the relative abundance of all the species that we see. And then we're also going to use this sample to do cell counts for our target species, except for Heterosigma and Pseudonychia. But all the other ones you would get from the net toe sample. And then we're going to archive it to save it with formalin and in a scintillation vial for the archive. For the whole water sample, you would use the live observation to look for Heterosigma cell counts when it's fresh, and any other cells that you see in there that that you might have missed in the net toe. And then we fix it with formalin, a specific volume of it, to then we let it sit for 24 hours, and then we concentrate it to 10x by removing some of the volume after the cells have settled to the bottom. And then what we have left is called the whole water 10x concentrated sample that we will archive and also use for Pseudonychia cell counts. So now to go into a little bit more detail about those processes. So first, for your net toe sample, you want to measure the volume with a graduated cylinder. Whenever you are transferring things from one volume to another, or like one container to another, like from the cod end to the container, or the container to the cylinder, and then back to the container, what you want to do is gently swirl to make sure things are like suspended and not sticking to the edges, but don't shake too vigorously, just like a gentle swirl will do. Just to make sure that like things aren't getting like stuck to the edges and then not being included in your observation. So measure the volume of the net toe and record that. And then the next process is the same for both the net toe and the whole water samples. Gently mix. If your lid is nice and tight, you can invert the whole container, or you can just gently swirl it. Then when it's well mixed and heterogeneous, you can pipette a small amount of water, it's 0.1 milliliters, onto your Palmer-Maloney slide. Put that slide under the microscope and identify organisms to the lowest taxonomic level possible. And then record everything that you see and record the relative abundance for all of the organisms that you can identify. And And there is an option for writing unidentified, and then again, describe it as much as possible. If it's a dinoflagellate or an unidentified pennate diatom, you can write that as well. But if you are able to identify it, you can do that. And then for the target species, we do ask that you do a cell count from one Palmer Maloney slide for our calculations.
We have a question that came in about what is meant by archiving the water sample. I'm guessing we're asking what we're doing with those too.
Great question. So formalin is a fixative, so it preserves the sample and the organisms and most of the organisms in it, so that if we wanted to go back and check, like reanalyze a sample, or kind of just like look for different species that might have been missed in the small sample that we take for the count, we have cells that are like preserved that we can look at. If you don't preserve them, things will die and disintegrate, and some organisms might eat others. And so that's why it's useful to be able to preserve them. Not everything is preserved equally with formalin, but formalin does a good job of preserving most of the things that we are focused on. It is something that we are kind of thinking and talking about as a program, because there are some usefulnesses for it. When you're looking under a microscope at 0.1 milliliters of even a concentrated sample, there's things that you could miss, even if you swirl it and mix it, you never know. And so if, for example, like a bloom happened and we wanted to go back and say, hey, like, let's try and assess like how quickly it came on. Let's look back at the last, 2 weeks before that. You have those samples saved. That's something that we could do. And also, if you have a lot of unidentified organisms and you're like, okay, now I want to go back, I have more practice, and I want to look at something and see if I can find that same weird organism again and identify it. That's another usefulness of these samples. And so that's why this program has historically been preserving samples, and we're continuing to do that for now. Some folks are less comfortable using formalin, and that's fair because it's a toxic chemical that's a carcinogen that we'll talk about a little bit more in a minute, actually on the next slide. And if you have concerns about that, you can feel free to reach out and we can talk about an alternative sampling plan. But the general protocol for most sites is to do the archiving.
And then are those just brought at the annual training every year, brought back to us and we store them in a refrigerator in our basement.
Yeah. Actually, formalin samples don't need to be refrigerated, but we do store them in a basement.
Oh, okay. Great.
Yeah. Yes. Yeah. Okay. Shall I move on?
Yeah. Thank you, Michelle.
Thanks for asking the questions. Okay, so yeah, record all the species that you find from your live observations. Oh, one more note on this. We do recommend doing live microscopy as soon as possible after you collect your sample. Like an hour, a couple hours or something isn't going to make a huge difference, but remember that when you use a net tow to concentrate everything, you're basically making like a buffet for the bigger things that you might catch in your net. And so if you wait too long, it can affect the balance of organisms in there. Also, some things just don't like being caught and will die, which can also be harder to identify if they're like breaking down. And finally, if you look at them live, it's the best because the way that phytoplankton move can be a really helpful indicator of what species you're looking at. Some of these organisms are quite hard to identify. The other thing is, we talked about this a little bit on Tuesday, if you were able to make it. If not, we'll hope to get the recording out soon. But the angle at which you're looking at a phytoplankton really matters when you're trying to identify it. And so if you have live samples, you're more likely to be able to see them turn or move to the right angle to be able to identify them and see the features that you wouldn't see, depending on how they fall. Which is a lot harder to do if they're fixed and not moving, because then you have to manipulate them yourself on the slide, which is really, really hard. So generally looking at them live is best. Sometimes they move a little bit too fast when they're alive, which can also be challenging. But again, observing the pattern in which they're moving can really help you. And sometimes if you watch the Zoomers like for a little bit, they'll pause, and maybe you can and see some of that, some of those features that help you identify them. So live observation is definitely best. However, in some cases, like, if you're in a crazy rush or something, and you have to look at the preserved samples, that again is another good way of, if you have them preserved, you might be missing some of the things that don't preserve as well, but you're still getting a good amount of our target species. So preserving samples with 20% formalin. So we use 20% neutrally buffered formalin as our like stock concentration, but we're putting a small amount into our, like the final concentration is less than 1% in our fixed sample. So the stock container you want to treat with caution. And I mean, you want to treat all of it with caution, but the dilution after it's been put into the seawater and diluted in there should be less than 1%. The main issue with formalin is that in the long term, it can be a carcinogen. You don't want it to contact your hands directly, and you also don't want to like inhale it too much. It has a really strong smell. So you want to make sure that you're using it in a very well ventilated area, and leaving containers open for like as short an amount of time as possible, especially the stock container. So make sure you have everything set up. When you open your stock container, take out the amount you need with a pipette and then immediately close that container again. We'll also recommend wearing gloves that are at least like 4 millimeters or thicker of nitrile to protect your hands. And then if you have an accident or spill, if it's small, and we only give you a small amount of formalin at a time, so hopefully you're never having like too large of a volume, but if you accidentally get it on you, just immediately wash it, throw out your gloves, and if any of your clothes get, if it gets on your clothes, take off your clothes immediately and wash them, and make sure you wash your skin underneath. So we do have safety data sheets available, and we have a standard operating procedure that we shared with folks last year and that we'll share with you all again. This is something that we will have hard copies of at the in-person meeting, and we'll also email out to everyone to have. And if we have time at the end of this meeting, I might be able to just drop them in the chat as well. Though it might take me a second to pull them up right now. So I'll keep going with the presentation for now, but I'll share them later. Does anyone have questions about formalin at this time?
We don't have a question about formalin, but we had a question about what needs to be recorded on the scintillation vial.
Yes. A very good question. I think that's on the next slide. I'll cover it in a minute.
Great. And then sorry, Stephanie, you might wanted to.
I was typing it, but how much volume is in the vial, and then how many drops of formalin would you add to that?
Oh, great question. The details are on the next slide. I think I said, do you have any questions about formalin too early? Because I actually have more information.
Okay, sorry about that. Thanks.
Yeah, totally. And then after this, hopefully this will answer your question. Okay, so the archiving procedure. So for the whole water sample, this is the one that has not been concentrated by a net. So the organisms in here are quite dilute usually. And so we concentrate this by 10 to be able to then look at things in a more concentrated way and also count the pseudonychia. So to do that, we give you a special 50 milliliter size tube. So you want to invert your 2-liter plastic bottle or gently mix it to make sure that nothing has settled down and it's all well mixed. And then you want to pour 50 milliliters into the glass test tube. Then you add 1 milliliter of formaldehyde fixative and then cap it and then invert it once to mix it to make sure the formalin is going through the entire sample equally. Then you want to let it sit for at least 24 hours without like shaking or jostling too much. And during this time, so when the formalin interacts with the phytoplankton, it kind of instantly kills them. And then these are really tiny things. So over 24 hours, they'll slowly settle to the bottom and concentrate at the bottom of the tube. So then after 24 hours, the next day sometime, what you can do is we have like long pipettes that we give you that you can use to remove the top 45 milliliters of liquid. And usually we say kind of go from the top, because you want to be careful in doing this to not kind of like squish any water back in, because that creates like turbulence that might mix the bottom. And you want to aspirate from the top, because if you shove your, pipette in too deep, you also might disturb the bottom. So basically you just want to like carefully take the top 45 milliliters of liquid off the bottom, and then you'll have 5 milliliters left with presumably all the dead phytoplankton kind of at the bottom. Then you want to gently swirl them so none of them stick to the bottom to like resuspend them, and then pour that into one of your scintillation vials. Then you'll label that with your site name, date, and WW10X to identify it as the whole water sample. And for your site name, a lot of you have like abbreviations that you might write, so you don't have to write out the entire name, as long as it's very clear and you use the same one consistently, that we know later if we see this like kind of 4-letter code, we know which site it goes with. And then all of our scintillation vials come in a tray. It's easiest if you can store them in the tray, just because it keeps them organized, and they have like little cardboard holders to keep them all in a row. And then you can just take the empty ones out and put the filled ones back into the same location. And then that toe sample is a similar process. You just want to invert it to make sure you've got a good mix of everything that's in there, and then pour about 20 milliliters into the scintillation vial. They contain 20 milliliters, so basically like near the top, but not totally full. These don't get frozen, so they're not going to expand, but just doesn't have to be all the way to the top. Then add 1 milliliter of fixative, cap that, invert it again so that it can evenly spread through the liquid, and then label the cap again with the site name or code, date, and write net instead of whole water 10X. Okay, so hopefully that answers all the questions about the specific archiving procedure. I'm not going to go into detail on this, but a couple of our sites do have additional samples that they collect. And these include collecting chlorophyll, or collecting on a filter, filtering water through a specific filter that can get analyzed for chlorophyll content. And then the liquid on the other side of that can be analyzed for nutrients such as nitrate, ammonium, phosphate, and silicic acid. And then on a separate filter, separating dissolved and particulate toxins for toxin analysis. The reason we don't do these at all sites is that these analyses take more specific instrumentation that we don't all have. And so we'll have to send those away to get analyzed, which costs money. So usually this is just for specific sites that do that are like kind of specifically designated in specific research projects. And if your site is one of the ones that does this, you also need a little bit of extra equipment, including like a pump that can take up a little bit more space. And also all these need to be stored in a freezer. So you also need access to a freezer. And I know a lot of sites are interested in getting additional information of total chlorophyll and nutrients in the water, just to like better understand your sites. And there's like there's other programs that measure chlorophyll and nutrients in the water. The Department of Ecology has like a route that they do. And I know King County and some of the county districts will do more sampling. So that kind of data from like a nearby site could be helpful. There's also some ways of measuring nitrate and phosphate with like these little kits. That is something that some folks have talked about before. We don't have any of this stuff kind of in place now, but for those of you who are interested in more information there, we can talk about it. Okay. So here's some specific instructions on how to use a Palmer-Maloney slide. So these slides are a really specific shape and size because of the size of organisms that we're looking at. If you use like a Sedgwick rafter, those are much larger, so they're better for kind of larger cells. And then there's also, hemocytometers, for those of you that might have a background in medicine, to count like blood cells, which are much tinier, so you might need like a much smaller one. For what we're looking at, which is organisms between 2 and like 200 microns, about 0.1 milliliters of water is a good amount to give us a representative idea of what's in the water without having to sit there counting like an entire giant slide. So that's why we have these really specific size slides. As always, I've repeated this multiple times. You want to make sure you're gently inverting or swirling your sample to mix it. And then pipette, get liquid into your pipette. And there's a couple of ways of filling your Palmer-Maloney slide. One way is to put the slide cover like at an angle and then start filling the liquid. And then because of surface tension, the water will kind of fill that space. The main thing you want to do is try to avoid air bubbles, because those will, one, affect the volume of your sample and also look crazy under the microscope. It sometimes will take a minute or two of practice if it's your first time doing it, but usually folks can get the hang of it after practicing a little bit. And then what you want to do is start at, there's several ways of doing this, but one way that I think is useful is start at a larger magnification, kind of scan the whole slide to get a good idea of what's in there, see if there's anything like large that might be moving around or moving other things around. Like if you have a copepod or something that's like swimming and moving everything else around, just like knowing that it's in there is helpful. And then you want to go at like a higher magnification and just kind of go through the slide, scanning it and counting and marking everything that you see. So while you're looking at your slide, what we're looking for most organisms is the relative abundance. The relative abundance is the measure of community composition. And so we're trying to kind of get an understanding of the biodiversity and composition in the system, which can tell us like a lot about how healthy the water is and the system. And this method is qualitative, not quantitative, but it's great to like very quickly analyze and record data, which is really helpful when you're doing it, every single week and asking lots of different folks to do it. If you had to count every single cell in your sample, it would take a lot longer. So we categorize things into 4 groups: absent if there's nothing there, present, common, or bloom. And the way we determine this, because again, this is qualitative, and so people often ask like, okay, so what exactly makes the difference between present and common or common and bloom? And so the way we try to explain it is bloom. Some other words you can use are prominent, common, or present, prominent, and dominant. So a bloom or a dominant organism is when you look at the slide and like that is like you're seeing it everywhere. There's like a lot of it. It's the most frequent thing that you're seeing. It's kind of like taking over the whole slide. That's what would be a bloom. If you're seeing the same thing multiple times, like it's definitely there, but it's not just like completely overwhelming the sample, then it's probably common. And for anything else that's there, but you're not seeing like a ton of it, that's present. If you see only one of a cell, it's present. And then to like ratchet up to those higher like abundances, it's kind of got to be like a lot more present. I know those aren't always satisfying ways of explaining that, but hopefully that is helpful. I have some pictures here that are kind of like a practice. So in this example, if you're looking at the different letters that are in this picture. There's a lot more A's than C's, B's, and D, B's, C's, and D's. So A would be like the blooming species. We're seeing like quite a lot of it in the slide. Then really none of the other ones are much more common. Like there's just a couple of all of the other ones. So honestly, we'd probably categorize all of B, C, and D as present and none of them as common in this example. I have some other pictures here that are not great. I think I wanted to update these this year and then forgot about it. So they're the same picture examples as last year, and as probably several years now if you've come to these a lot. But to give you an example, in this case on the left, what we're seeing here in these long lines are pseudonychia, and there's quite a few of them, but they're not overwhelming the whole sample, so that's what we would call common. And then these other species, Serratium, Thalassiocyra, Chaetoceros, Didylum, and all the other species that are here are only categorized as present because none of them are super, super common. The other key here is that, we're comparing things of very different sizes. So that's something to keep in mind as well. You're seeing 3 of these larger Serratium species. Though they're taking up more space on the slide, they're bigger, but there's fewer of them. So kind of other things to keep in mind while you're categorizing these. And in this slide, the Rhizosolenia would be considered common because we're seeing quite a few of them around, but all the other species would be considered present because there's fewer of them. I know this has been a question in the past, and I hope to get some better kind of explanations and pictures, but hopefully this was helpful, and definitely open to talking about this a little bit more if people have some more thoughts or questions.
We did have a question come in about if you need to see more than one slide to decide if something is common or not. And yeah.
No, so the idea behind swirling is to make sure that you're having a well mixed from your cod end to your slide, and usually we'll do our kind of main analysis off just one slide. If you have more time or are curious, you can look at more slides if you want. And it is interesting to see how much variability you see between those different slides. For things that are present, sometimes you'll miss them on just one slide, right? We're going from the whole water in the ocean at your site to kind of what's concentrated in one small area. The vertical net toe is trying to get at least the depth averaged kind of composition, but you're still only looking at one spot, right, out of the whole water. And then you're taking all that, swirling it down and taking 0.1 milliliters and seeing what's in there. So there are things you're going to miss. So if you look at more slides, you are going to see more diversity. But the idea is that by making sure it's swirled and well mixed, you're still getting an idea of what's there. And if you're having, if there's a bloom in the water, you're going to see that in your slide if you collected your sample correctly. And if things are common, you're also going to see that in your slide. If things are present, there might only be a couple there. And so you might see them on one side and not another, which means that they're less important to the whole system, right? So if you looked at, 10 slides, you might see one Alexandrium cell, and that's worth being oh, they're here, but they're obviously not so abundant that they showed up in every single slide. So the amount of concern is lower.
And I think the question they meant more of in one view on the slide instead of just
Oh,
the whole
yes.
slide. So yeah, you got to do the whole slide. But
Apologies.
no,
I
no,
thought
no.
you meant,
The way I phrased it was
yeah. Yes,
so,
these are just examples that are just pictures that somebody took a long time ago. I don't even know who. But if you are doing this, you would be looking at your whole slide. This is why it's helpful to scan your whole slide at a lower magnification first, because then you're kind of getting an idea, and then getting a better idea of what's really there when you kind of go in at a higher magnification. And what different folks have different ways of doing this. I'll actually be curious for folks that have been doing it for a long time, kind of how you do it. But on our data sheets, we have a bunch of species listed. And as you're like going through, you can like mark when you see them. And then if you like start looking at a slide and you're like, okay, this one, some people tally or some people just like write down the names on a piece of paper. And then as you're going, you're like, okay, I'm actually seeing this one like reappear a lot. I'm going to boost this from present to common. And then things that are bloom, you should probably like know like kind of pretty immediately when you're looking at your slide that like, okay, this isn't every single view, no matter where I go, they're like kind of dominating the sample. So yeah, that's how we do it. Hopefully that helps. But yeah, it is based on the entire one of a Palmer Maloney slide. So not just a single view, but the whole slide.
Yeah, that's great. And then do you mind going over the right photo and saying which ones you would say are common or just present?
Yes. Rhizoselenia, so that's this this pointy long one with the points. We're seeing it here, 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 and here, even though it's kind of fuzzy. That's several cells. So, and again, this is based on just this picture. In this view, assuming the rest of the slide looks like this, we'd probably be like, oh, it's probably common because we're seeing it quite a lot. Let's see what else is here. So these notes, again, this slide is an old one taken from a long time ago. So the other things they have listed as present are Didyllum, Pseudonychia, Chaetoceros, and Hemialis. And so I'm like looking to see. We've got like some Chaetoceros bits here and here. Honestly, it's not a great picture, so it's a bit hard to tell. Hemialis. I don't actually see the Didyllum, and I'm not sure which ones are the Pseudonychia. So really, these are not great example pictures, and so I apologize for that. So perhaps maybe next year, I'll remember to replace these with some better pictures that we find.
Maybe we can send out some guides for just like, I think there's some folks that are having consistency within their groups. So maybe we can send out some more examples of like what we're defining as common and present.
Yes. What would also
thanks for that comment, Paula.
Yeah, what will also be helpful is if, we often get pictures of individual cells being like, what is this specific cell? But if you take pictures of like more zoomed out versions that have like a lot of cells inside, that can be helpful in helping us determine, like we can kind of look through those together and cross-check and make sure we're all like kind of going along the same guidelines of what's common and present. Yeah, that's one upgrade that I think I meant to do last year. And of course, here we are, haven't done it, but well, let's work on that before next year, in the next year. Okay, oh, wait, is everyone okay to move on from this? I know this is like a sticking point. Yeah. Okay, we'll move on for now. I apologize. I was out sick last week. That's why I keep coughing. And so I didn't have as much time to edit this before this meeting as I had meant to. But thank you everyone for the questions and the comments, and we're gonna try to incrementally improve things. Okay, so okay, for our calculation, so for our target species, in addition to writing down the relative abundance, we ask for you to write down or count how many species that you're seeing. And again, it's important to know the volume of the water filtered into the cot end and the amount that you towed. And we have spreadsheets available that we'll share with you if you don't have them yet, but hopefully you, for those of you that are part of the program, you have access to them that help you do this calculation quickly. But I do like to kind of show where the calculation comes from, just so folks can understand that if they want to. So do not worry, you do not have to do this math every single time, but if you are curious as to why we do what we do, the top of the net is a circle. And if you tow it through water in an ideal system where the water is not moving and you're moving at a steady pace, you're in theory filtering a cylinder of water into your cod end. And so by knowing the area of your circle and the depth or the tow length, you can calculate the volume of that cylinder, and that's how much water was filtered. So again, knowing the net diameter, the number of toes to get the total toe length. Sorry, I've put so many animations into this. I think I clicked them all now. So in theory, you can find, you can multiply to find the area of your circle and then multiply by your toe length and number of total length by the length and the number of toes to get the total volume of your cylinder filtered to calculate how much volume is filtered. Okay. Then to get from your water column and all the cells that are in it, you filter that down into a smaller cod end that has all the cells in it, and then you take a small amount of that out into your Palmer-Maloney slide. So using this equation, you can find the concentration of cells in the water by multiplying back out, right? So you're only looking at 0.1 milliliters if you count 10 cells in 0.1 milliliters, but multiply it by like the volume of the cod ends, then you know how many cells were in the entire cod end. And then if you multiply that by your volume filtered, or divide that by your volume filtered, then you get the total number of cells per liter in your water system. So again, we have, so you don't have to do this math all the time. We have some really nice Excel spreadsheets where you only have to fill in the parameters that pertain to you, the area of your net. Most people should have the same size net, though there might be some variety. Then you shouldn't have to change that after you've set it once. Your distance towed, you'll change every single time. For the cod end volume, you can change that as well. And then the volume of the Palmer-Maloney slide should also not change. They should all be 0.1 milliliters. I think there are some that are like not official Palmer-Maloney slides that might be slightly different. But in general, they should all be 0.1, but it should be written on the slide case or cover when you received it. And then you should only have to edit the number of slides, number of cells that you count. And then it'll calculate it all for you. For the 10x whole water calculation, an easy way to do this is to just add 3 zeros to the end of whatever cell count you got. But if you want to be sure and double check, or if your Palmer-Maloney slide is a different volume, then you can type it into the spreadsheet and get your answer. Okay. All right, so now we're going to talk a little bit about our target species in Washington. And we are going to talk about this a lot more on our in-person training day on the 18th. So there are 3 main human health hazard HABs that we look out for. Alexandrium, I've already mentioned this one, creates paralytic shellfish toxins and is pictured right here. We're going to talk a lot more about the identification, but they often are chain-forming brownish dinoflagellates that sometimes look like little hamburgers. Pseudonychia, this is a diatom that also forms chains. It's like a long, thin species that kind of connects to each other and makes like these long chains. And it creates domoic acid, which causes amnesic shellfish poisoning. And then Dinophysis, there is a lot of morphological variety in this genus. So there's a lot of different species to look out for, and there are ways of identifying them between each other. And they create diuretic shellfish toxins. And different species might have like be more potent and not. And so identifying them to the species level can be helpful in, estimating risk. We also look for Heterosigma akashiwo. This is a species that causes fish kills, especially dangerous for like penned fish that can't swim away and can cause some pretty big problems when it blooms. We're also looking for 3 species that are related to shellfish mortalities, Akashiwo sanguinea, Phaeocystis globosa, and Protoceratium reticulatum, which are pictured here. We're going to talk a little bit more in depth about identifying these, so I'm not going to spend too much time on it today. But I did want to let you all know, we're going to talk about this more in the coming trainings as well, that we have 2 new target species that we're looking out for this year. In the past, these have been categorized as like emergent threats or like things to look out for, but that weren't required or mandatory reporting. But now we are going to focus on these this year. And so we're going to talk a lot more about them in our training. But Azodinium, this is a species that we know, this is a genus that's been seen in the Puget Sound before. And in fact, a couple of years ago, I think our program helped with some research to identify different species in the Puget Sound. This is before I worked here, but so we've known these have been around for a long time, but they create toxins that can affect human health. And so there's something that we're looking out for. And we'll talk a little bit more about this coming up. And then the other group is the Karenia genus. The one we've in the past told you to look out for is Mikimotoi, which is pictured here. The ORHAB partnership, our like sister phytoplankton program in the Olympic region, has seen a lot of Karenia and other like dinoflagellate species in the unarmored dinoflagellate species on the coast this past summer. And these also are concerning because different species create different toxins. So Karenia brevis is the one that is kind of the most infamous, I'll say, for like Florida red tides. And some of the other species in the genus might create similar or slightly different toxins, but we're definitely concerned about them. So we want everyone to look out for them and know how to identify them. So we are going to talk a lot more about them on the 18th. So we hope you'll be able to join us for that to learn how to identify them. Okay.
We have some questions coming up in the chat about not about the new target species, but about why a cod end is called a cod end. And I don't know. So if you have any,
I don't know off the top of my head. But
we got a
can
reply from someone else that it comes from the end of a fishing trawl. Like it's where the cod get caught. So I don't know. Yeah, seems right. But I don't know the answer to that one.
I believe it. Thanks for answering that question. Yeah.
Oh, that was Alicia, I believe I answered.
Oh, great. Thank you. I learned something new too.
Okay.
Awesome. we do still have our, you know, kind of additional concern species. These are kind of less frequently problems in our area, or kind of not common yet, but we ask you to look out for them. So Chytoceras concavicornis, this is one species of Chytoceras that has on their setae, they have like barbs that can get caught in fish gills. So they're also like a fish killing species. So that's one that to look out for. Margolefidinium, we have this, I feel like fairly frequently present, sometimes common and blooming in several of our sites in Puget Sound, but and this species can cause problems in like other areas of the world. It's like a huge problem for fish kills. I think it's been kind of like less frequently a problem in our area, even though we see it quite frequently. It doesn't cause the kind of environmental damage. Maybe the blooms are just kind of smaller and more contained, but still worth like looking out for and paying attention to. One thing people also often say is that like HABs are all HABs are local. So it's like kind of how that species affects your ecosystem and where you are in your system. So some organisms that kind of are huge problems in other areas might be present in ours, but not as might not cause as many problems. But that being said, with like environmental change and changing conditions and species adaptations, things can change. It's not always going to be the way it always was. And so some of these we say like, you know, look out for these, because if you are starting to see them in a large abundance, and we're also noticing fish kills or shellfish mortalities or other problems, you know, then that's something we'll start paying attention to more. Lingulolix polyedra is one, another species. This was recently renamed from Lingulodinium, which is how you might have heard it in the past. This one has also caused a lot of problems in like Southern California. I think they see it pretty frequently on the coast. We don't see it as frequently in inland waters, but I think it creates SoundToxins like Protosteracium reticulatum. So if we do start seeing a lot of it, it could be problematic for shellfish. And the Mesodynium rubrum by itself is not a phytoplankton that causes too many problems, even though it can bloom pretty thickly and cause the water to turn red. But the reason we look out for it is because it's a particular prey species of Dinophysis, which is a problem here. So if you see a lot of them, they could then lead to an increase in Dinophysis. So good to be aware of. Okay, so the next important note is that for chains and colonies, you want to count every cell you see, not just every group. So for example, for Pseudonychia, this would be 4 cells. And for this Alexandrium, it would be, spoiler alert, 13 cells. The exception to this is for Phaeocystis. That's one of our target species that they're like tiny flagellates that form colonies in kind of like these mucus matrices. And these are extremely hard to count every single cell of, just because sometimes they're like overlapping in like a 3-dimensional gel matrix, and quite tiny. And so for those, and for the project that we're like working on, just like estimating the relative abundance, and then if you can count like the number of globs you see, that kind of gives us an idea of how many there might be. Okay, so for alerts, when you look at your sample and you might see some target species, to report an alert, you would email soundtox@uw.edu. Our guidelines for this are to report like the first time you see Alexandrium and Dinophysis, especially if you haven't seen them for like the first time of the season, but also if you then haven't seen any for like, 2 months or a couple of weeks, and then you're seeing Alexandrium again, let us know. Both of those are, can cause problems at lower abundances, so it's good to know about. For most of the rest of the species, Pseudonychia, Heterosigma, Akashiwo, Phaeocystis, and Protosteracium, we would like you to report when the relative abundance is common or blooming. Again, if you just see like 1 or 2, not as big of a deal. If you are seeing kind of like a numerous amount, enough that you're seeing like several on like every slide or every view of one slide, that's when you would start reporting those. New of this year, if you see Azadinium or Karenia at all, please let us know. We're going to work on figuring out what levels over the next, year to couple of years, depending how often we see it. We're going to figure out what the right level is to report and how concerned we need to be. If possible, please include the concentration in cells per liter that you would have gotten by calculating with the spreadsheet in your email to let us know how many you've seen. But if you are seeing something concerning like Alexandrium in the water or dinophysis, then it's helpful to send the alerts as soon as possible, and then you can always like follow up later with the concentration. So if you're like, oh, I'm just looking at my slide quickly and I'm seeing quite a bit of Alexandrium, I'll send you the details later. That's also helpful. We do have an alerts key, and I did not have a chance to update this yet. So Azadinium and Karenia are not on here, but I will update this before our next meeting and email it out to folks just so that we know the new species are on there and important. So the 2025 alert key will come out. But this is basically just summarizing the information, reminding you where to do the counts from, and any specifics for that species. So, for Heterosigma, if you want to go from the whole water sample, from Pseudonychia, whole water 10x, for Dinophysis, if it's possible to distinguish the species, please let us know, and it gives you when to alert. So that is our basic protocol up until data entry. So if there's any questions about that, and then we can go into the database next, if you don't have any questions. Okay, great. So we have a database that's used to enter HAB species. We have a database to store our data. It's helpful to enter HAB species as soon as possible because the Department of Health is constantly looking at the maps, especially for information about how to prioritize their biotoxin samples. The alerts are really helpful to call attention to kind of new information, but they have said that they use these maps really like daily to help with their biotoxin prioritization. So to access the database, it looks like this when you log in. If you have any questions about getting, a password, or if you have trouble logging in, just email us at soundtalks@uw.edu. When you go to sign in, our data policy will be pasted there, and by signing in, you are like agreeing to adhere by that data policy. So our data policy includes, first of all, it's a private database for just partner access, except for, well, DOH being a partner has constant access for like public health and safety reasons. The reason our data is so protected is to maintain trust within our partnership, including to honor Indigenous data sovereignty and also for the privacy of industry partners. So we have a variety of different partners, including community scientists and aquaculture businesses and native tribes. And so we want to make sure that everyone is comfortable with how their data is being used. So when you sign in, the data policy that you like agree to is that being aware that it is a editable kind of changing system. So data is subject to change. If you have access, you should not share that access with anyone else that's not part of the partnership. The data that's entered and collected is like privately collected by partner use only. And if you would like to use the data for any analysis or anything, please reach out so we can make sure we have all the correct permissions from all the other sites whose data you would use, and to make sure that it's like an acceptable use of the data. And so by signing in, you agree to all those terms. Logging into the database. So a quick way to do it is go to the SoundToxin website. and click on the data tab, which brings you straight to the page, or you can save the this address that to just go straight there. And then each person should have their own username and password to sign in. Again, by signing in, you agree to the user agreements. When you first sign in, the first site that you will, or the first page that you have access to is the sites page, which will have at the top all of the sites that you have edit access to. So if you only monitor one site, it'll just be up at the top, and then everyone else's site will be below, but without the option to edit that location. So for your site, if you're adding some new information, you would click on the add update or add/update visits. And then you can click on new sites, new visits to start a new visit, new data entry. So when you create a new data entry, start by filling out the sampling date and time. By clicking on the icon, which will give you a drop-down calendar and have a spot for you to change the time as well. And then when you're done, click done. And then as soon as you've inputted collected by and the sampling date and time, you can click create, and then then it'll like kind of create the new sampling visit entry. Then you can fill out the rest of these like environmental parameters that are on the first site, which is including air temperature, water temperature, and salinity in degrees Celsius, and using the drop down to change the weather, the tide, and the winds. You also can fill out how many meters you towed and your cod end volume here. If you are a regular site, most sites, you can just ignore the rest of these entries. These are kind of like, they're for the specific sites that are collecting nutrient, chlorophyll, particulate, and dissolved toxin data. You would fill in these four, and then these are for the like final results that we can add later, so you don't need to fill out anything else if you don't want to. But please do feel free to fill the comment section with as many comments as you would like that seem relevant, especially as it relates to the condition of the water or the sampling event. I mentioned earlier, like if there's a super strong current or something and you see your net kind of going sideways as you're pulling it up, put that in the comments. It's helpful to know. If you notice like dead fish or animals or something, or that all is important data when it comes to phytoplankton data, because fish kills and shellfish mortalities are part of what we're looking for. If you're seeing visible discoloration of the water, like that, all that information can be really helpful. And if you want to include any extra information, like if you do collect the wind speed, not just how like kind of moderate or whatever, you can put that information in there as well. Like it's always great to have extra information, even if we don't have it as a specific field. The next section below it on the data entry site is called our required observations. So in this section, you are required to fill in the relative abundance of the target species. So at the moment, and probably for the next month or so, it'll look just like this. And then we're hoping to add Azadirachta and Carenia at some point to this section. If none were seen, if you fill in absent, that's helpful for us later when we analyze the data. If you just leave it blank, it's just a little like, that's like, we're like, was there something or not? But if you don't see it in your slide, it's okay to mark it as absent. Then if you did observe cells and it's present or common or blooming, then you can click the cell counts to add additional information. If you filled out all the relative abundances and then you want to go in and add the cell counts, make sure you just go to the bottom of the page and hit save. Any changes that you make without hitting save will not be there if you navigate away from the page and come back. So you'll have to fill it out again. So saving in between is very helpful. And then for when you click cell counts, each species of the target species will come up with a little page similar to this, where you can fill out what species you saw, the cell counts, the water source, and any additional comments that you might have. You can ignore the perk species on the Alexandrian page, but just put in the comments if you see multiple species.
You also want people, if they have multiple people collecting samples and counting plankton at multiple sites, do you want them to identify those people in the comments?
Yes, thank you for bringing that up. I meant to say
That's
that.
from Jackie.
Yeah, thank you. Yeah, so you might have noticed that when you put in the information here, there's only one person in the like collected by dropdown section. And these data entries, especially for volunteers, also kind of help us track who's working on what. And so even if not, it's helpful to know who to go back and ask the questions to, like for all partners, for any monitor who's monitoring. If we're like, I have a question about, what you saw and I want more information, like if you list everyone's names in the comments, that's helpful, because often it's not just, in some cases, it's just one person that does everything. In some cases, there's a lot of people working on this. So we'd love to make sure we have everyone's names recorded. So great catch, thank you. Where was I? Oh yeah, so fill in your cell counts and your water source and the species if you know it, and then hit save, and then you can hit back to return to the previous page, like that. And then there are some specific, for Dynaphysis, we ask that's one where we ask that you mark or like record how many different species you're seeing and the percentages. For Pseudonychia, similarly, we ask that, but rather than species, it's morphotypes, which is just large and small. So for Dynaphysis, your page will look like this. If you're seeing multiple species of Dynaphysis, we recommend putting SPP here, and then in the species section, you can add what the specific species that you saw and just the rough percentage of how many you saw. So in total, you saw 10 cells per liter of Dynaphysis, and 4 of them were Acuminata and 6 of them were Acuta. You can put that in here. So then we can know the percentages as well. And then for Pseudonychia, what it looks like is the percent of the small and percent of the large. So a slightly different way of recording it, but just write like 50-50 or 20-40 or however you see these. And we'll go into details on Dynaphysis species and Pseudonychia morphotypes at the next training session. Okay, the last category on the first page is bloom observations. And so if there was one species that was completely dominating the slide, we ask you to report it in the bloom section. Sometimes there are multiple species blooming at the same time, and you have like maybe 2 species that are both kind of equally taking up space on your slide. For this section of the database, just pick one of them. And if the thing that is blooming and taking up all the space on your slide is one of the target species and you've already recorded it as blooming, then you can just not record it again, because the system doesn't like it if you have 2 things that have the exact same description. That can get confusing. So if you've already marked it as blooming, then just leave this part blank or put in none, and then that'll be like removed from any of our analyses.
We have a question from Leslie about if we need to do, are cell counts necessary on each Dynaphysis species seen?
Not on each species. So if you're seeing a bunch and you're going through and counting every single one, and then you have a total number, that's fine. And then you can like roughly estimate the percentages, if that's helpful. Like, so if it's, say rather than 10, you counted like a lot more, and you're like, well, I didn't actually specify which one was which, but I know about half of them were this one and about half were that. That's how it's recorded in the database. So if it's not perfect, it's okay. But if it helps you to while you're counting to do that, that's also fine.
Thanks.
And then the last section is to fill out in the database is the additional observation section. So scrolling back up on the page to the required observation section, there's a little box underneath that says additional observations. So make sure you save your page before you navigate to this to make sure you don't lose anything that you filled in on the main page. But if you navigate to the additional observations page, this is where you can add in all those non-harmful species that you took the time to identify. So here, it'll list, it'll just be like, oh, you can add as many rows as you want by either clicking add row here at the top, or if you're typing quickly, if you hit tab, it'll switch you from column to column. And if you go all the way to the end and hit tab again, it automatically makes a new row. So if you're the type of person that likes the keyboard shortcuts, you can just hit tab several times to get to a new row. You can also click and use a drop-down menu to find the genus and species that you want, or you can start typing. And if you start typing like CHA, it'll like jump to that spot in the drop-down menu. And then you can also use your arrow keys to go up and down. So especially like when I'm filling out data entry and I start writing THA for Thalassiocyra, Thalassionema also comes out. So sometimes you just use the arrow key to like get to the other one. Hitting enter also will like select it and then pop you to the next column. So you can also use the drop down there to get the species if you know it. And if you don't, that's okay. And you can also add the abundance there. So those are just some keyboard shortcuts to go faster. If you've accidentally made too many rows or like put the same thing in twice, you can click the 3 lines to delete the row. And again, I think the thing that is the things to be weary of is that if you have 2 things that are the same, the system will give you an error. So if you ever get an error message, you can take a screenshot of it and send it to us, and we can try and help you kind of figure out what's going on with the database. But I find that a frequent one is if you've kind of duplicated. So for example, if you've accidentally written like Protoceratium reticulatum, which has already been recorded in the required session in the additional species section, it'll be like, wait, this is a duplicate. We have already seen this one. So just be aware of that. And in the drop-down menu, there's also a few other non-phytoplankton things, including like eggs or copepods or barnacle larvae and a couple of other things that you can add in there if you see them. We also have a couple of like groups. So I think like all eulinoids are in one group, all coccolithophores are in one group, rather than being divided into genus. And there's also unidentified options, so you can put that in there too. If you see something but you don't know what it is, you can record that. Always save before going back to navigate to the other page. I don't know if there was a difference between these two. Oh, add row, 3 lines to delete, and then always save, and then go back. Okay, click save one last time before you navigate away, and then you're all done. If you ever want to go in and add just your required species and then type in all your other species later, you can always just save whenever in that process and leave and then come back. If you want to go and edit an entry you've already filled in on your visits page rather than clicking new visit, just click right on the date and time, and it'll reopen that visit, and then you can go in and edit anything that you need to. There's some other features of our database that you can use if you want to explore. Under the visits, what you're seeing is basically a recording of all the like environmental data. If you click My Visits, it's just the sites that you have access to. All visits is the whole network. For observations, you're instead of looking at one row of data for every sampling event, you're looking at one row of data for every different observation. So you're going to have, say you saw 10 different species on one day, they're all going to have the same date, but all different species. If you want to, download and analyze or manipulate your own data, you can do that from the My Visits or My Observations pages, and we can help you. I don't have all the specific details here, but if that's something you're interested in, reach out and we can help explain to you how to do that. You can get a CSV file of your data and then you can analyze it however you like. And then the next most important tab that is useful to look at is the maps page. That's the one that DOH uses a lot, but you might also find interesting. It gives you kind of like a color-coded map of what's going on in the area, with green sites having reported no harmful species, gray sites have not reported in the kind of length of time, a recent length of time. Yellow means there's some presence, which the amount depends on different species, and then red means, oh, there's a problem here. And so it's kind of nice to be able to look at what's going on your neighboring sites, just to like help understand the site that you monitor. That's about all I have to talk about, but if you have any questions or issues with the database, just reach out to us and let us know, and we'll try to help you navigate that. And that's all for all of our protocols today. So this is my thank you slide, but also time for questions if anyone has some. All right. Don't think there's any questions. So that's good. Okay, well, we are going to send out additional resources that we mentioned here. Oh, the other thing I should mention is we do have a listserv that we send out information on, and in some cases, especially for groups that have many monitors for one site, there's kind of like one point person who's probably on our listserv. But if there's many people that go at that site, if you would like to get additional folks onto the listserv and you might not be getting the messages directly, like just reach out to us and we can add you to that. And also thank you to the folks who kind of send information to their groups as well. That's helpful. But yeah, if you'd like to join the listserv, let us know. We can send out resources through there. And yes.
We also we had just a quick question about where can we get the online version of the protocol manual, and it is on our website, but we can also email it directly to you. So Jennifer, I could send it to you, but it is kind of deep in the website as well. Or it might be on the first page.
We also are in the process of trying to make our website a little bit more easy to navigate and up to date. So those changes will be coming soon, hopefully. If it's hard for you to find stuff, we apologize, but yeah, we can try. I was gonna say, I think I said I was gonna share some things in the chat. If you guys want that, let me know and I can look those up now. And if not, we'll send out kind of like a list of things kind of maybe after all of our trainings.
Yeah, and we can send out those formalin kind of like SOPs and reminders to everyone, probably. After our training in person.
Yeah, maybe after February 18th training. If you want it sooner, let us know, but maybe we'll just kind of make sure we have all of our resources and blast them all out all at once.